that's for like LB,
It's already
LBA,
recording.
LBS.
Hi, yes. She's about to laugh when she sees your house. Ay, Dios mío.
All contribute, like you take some and I'll take two or something, you take two or something, so that when she does come back, she has results, even if it's good.
How do you feel about your notebooks? Better? The note she did, yo, I took it.
This one is the plates on this end is one per pair. So if you.
Well, at least it's getting there.
Other one.
I don't know how you do it.
I'm barely doing it. And then I'm passing my AEP class with the AEP.
That's good. Remember, you just, to know your actual grade, you just add both of the grades and divide by
He
2.
split it. He added our, supposed to took our grade from lab,
And they put it in,
put it in.
they didn't do that for us, right? Yeah, she already has her full grade
So
in there.
I'm at
We didn't have that
88
shit.
now. So I'm at 88 now.
Also, all her questions and her quizzes have only been 10 questions.
Oh, her quizzes?
Yeah, for A&P. A&P 1 and 2. For A&P 1, we didn't have 10 questions for our quizzes. We had like 50 and then 100
Yeah,
after.
we had a lot of
All her tests too aren't models, they're pictures on paper. We didn't have that shit either.
They're all people. And they're only 10 questions.
Good.
What?
But our practicals,
So
I
Mariana,
guarantee you, are a lot bigger than the exam.
this half is one per person. She said that she hasn't decided whether she's gonna move forward and do it for her, or if she's gonna have her come in and do her own half and then move forward. This half is one per group, so you would already have the results from when she comes back. You would just have to explain it to her.
Okay.
So this
So
is the only
basically-
one that she's, I guess, worried about because that is
So
this side is
so
just
basically, I'm grabbing one of everything.
one of everything, yeah. And then me and her are grabbing one of everything on this side, and then only one
Can
for
you
us.
guys finish? Can you guys help me?
Yeah.
You guys.
No, I'm out, my nigga.
Ay, ay, ay. I'm gonna go.
Sometimes they're not all. Are they all? Yeah. Okay, great. The one that is like on that side is one per person. This side here for conjugation is one per family. TSA. So for the plates on the left-hand side for enteric, you can abbreviate. It's like TSA, SS, HE, MAC. Oh, then I wonder if I didn't enter, I wonder if my own size. Thanks for reminding me, I'll check.
Yeah, thank you.
Everyone settled, we're gonna start with congregation.
I meant to bring the fan too and I didn't.
Okay, so as everyone is getting organized, if you could, we're gonna start with conjugation, I'm gonna kind of hide the bottom so that we kind of separate the two. But if you could pull out, you have 5 like yellow plates, LB, 2 LBs, an LB ampicillin, an LB strep, and then an LB with amp and strep. And the rest of the stuff you get set aside. Okay, so conjugation is one of 3 methods of what? Cover this, I think it's picture right now, right? Huh?
Reproduction?
Not reproduction. What's the method by which bacteria can transfer DNA from one cell to another? Horizontal gene transfer. Remember, it's conjugation, transformation, and transduction. Okay? So we are doing conjugation now. In a week, I think, we do transformation. We don't do transduction. But conjugation is the form of horizontal gene transfer that requires direct contact between 2 cells. The other methods do not. They're different. Right? So you have one cell that we call F plus or F positive. And by that, I mean that this cell has an F plasmid. That F plasmid, the F stands for fertility, and it's related to conjugation. So on that plasmid are all the genes related to conjugation. So think of it as, for example, the gene that codes for this F pilus. The F pilus has many names. F pilus, fertility tube, conjugation tube, sex pilus, they're all the same thing. Okay? So on this cell, if it's F positive or F plus, that means that it has an F plasmid, and so it has all the genes required to initiate conjugation. So this cell can initiate conjugation but that other cell has to, and that means that that cell lacks the F plasmid. Okay, so you never have conjugation between 2 F plus. It has to be F plus and F minus. Now, it initiates contact with the cell through the F pilus. That F pilus then retracts and pulls the cell closer to it, and then this forms this little tube through which it can transfer half of this DNA molecule, so one strand. One strand is fed through here. As it comes out the other side, there's a DNA polymerase that is making the complementary strand. At the same time, there's a DNA polymerase here replacing the strand that was transferred over. So that in the end, you have 2 F positive cells. Both of them have a double-stranded DNA molecule, double-stranded F plasmid. You never really have single-stranded DNA, okay? So when I draw this here, it is double-stranded. This one was the cell, imagine, that started with the F plasmid. This one was the one that received a copy of it. Okay? Now this cell too can initiate conjugation with another cell. In this experiment, we are going to utilize a cell line that contains an F plasmid, so it's F positive, and on that F plasmid, there's an antibiotic resistance gene. That's not normally the case. Those antibiotic resistance genes start on an R plasmid, resistance plasmid. But in this case, imagine on the F positive cell, you have an antibiotic resistance gene. So when that F plasmid transfers, so too does the antibiotic resistance gene. This is where it gets really dangerous, because any F positive cell can initiate conjugation with any bacteria. It does not have to be the same species. And then as it's transferring the F plasmid, it's also transferring that antibiotic resistance gene. For the sake of this experiment, imagine that the F positive cell has an antibiotic resistance gene on its F plasmid. The F minus cell, imagine it has a different antibiotic resistance gene on its chromosome. So when conjugation is complete, we create this new strain that has 2 antibiotic resistance genes now. One that it acquired through that F plasmid that transferred over, and one that it started with on its chromosome. And so this is what we're going to look for in the very end to confirm whether conjugation occurred. In the very end, when we're done with this experiment in a couple days' time, will we have this cell line that has 2 antibiotic resistance genes? meaning that if we prepared media with both, say, ampicillin and streptomycin, we could still see growth of this new strain of bacteria. Okay? So on your bench, you have 2 strains of E. coli. One has the ampicillin resistance gene, one has the streptomycin resistance gene. We don't yet know which one's F positive, which one's F negative. I'll just tell you that one of these is on the F plasmid of one cell, the other one is on the chromosome of the other cell. Okay. Part 1 of conjugation is that we want to determine which strain has which antibiotic resistant gene. We can't yet tell which one is F positive or F negative. We just want to know, is strain 1 the one with ampicillin resistance gene or streptomycin resistance gene? Now recall that if a cell line has a resistance gene, that means it can grow in the presence of that antibiotic. So we will start with these 4 plates, LB, LB ampicillin, LB streptomycin, and then LB ampicillin and streptomycin. You're going to divide each in half on the underside. Label one as 1 for strain 1, the other side is 2 for strain 2. You're going to grab some inoculum of strain 1 and just do a single line. Sterilize your loop, grab some inoculum of strain 2, do a single line. Do that for all 4 plates. And then we can talk about this more on Thursday, but you can imagine that if strain 1 has the ampicillin resistance gene, you would see its growth here, but not of strain 2.
There's 2 of
Okay.
these.
So that's what we'll be looking for on Thursday. So go ahead and start with this.
You have 4 plates or 5 plates? I have 4. And then I think this one was on that. Was it on that side? I'm gonna go check.
No, that's the clear one.
Oh, the clear ones? Oh, I have 5.
You have 2 LBs?
Yeah.
Why are they 2 LBs?
I don't know. I'm about to go check because I got the news.
Yeah, why are there 2 LB plates?
Why are there 2? One we're reserving for this, and I haven't gotten to that yet.
Thank you.
Yeah. So put one aside.
Okay, put one aside.
It might not matter.
So we color on one side, color to on the other side.
Where do you write it?
To the side.
We gotta draw a line down the
No, you're missing one. You should have an LD amp and strep.
This one? Of the 8?
That's TSA. You should have 4. Let me go get it. Here, go ahead and start. Go ahead.
This is what we do on one side. So I just didn't know we were using those for these 2.
Thank you.
Yep.
Yeah, LBAS.
And you draw a line
Yeah.
and then I pull out 1 on one side, I pull out 2 on the other.
E. coli 1. E. coli 2. Yeah, I'm labeling should put that.
Allow the plates to sit. Then draw a line down the middle each of the
Mariana wants to see. Yaz wants to see what Mariana's doing.
Hi, mommy, you left me.
There you go.
obtain the fluoride treatment 1 and 2 liquid culture that will be shared between your group and one other.
So they will label E. coli 1 on one side, E. coli 2 on the other side.
So,
I miss you for this.
Turn it down a little.
Oh, you're just scared.
And they're both with the loop.
yeah,
I don't have.
because any broth is, I'm not really.
I've been taking things out of Yaz's drawer, but like the matches, the inoculating loop, and the needle, everything in my drawer is missing.
Everything was missing?
Yeah.
And you have,
That was from her, yeah.
do you also need
Yeah,
matches,
yeah.
Jane?
I'm just gonna give you one.
Okay, I'm pretty sure I do.
I just wanted to let you know just in case of when. I also took her crayon thing because mine was missing.
Okay. You guys are good.
So when I pass it to you, you can do 2.
That's me too. We're not doing nothing with the water?
I don't think so.
No, I read it.
So whenever
I
you-
did
Do you
read
want
it
help?
correctly, right? You just,
I think I got it. I like doing this part.
Okay. Speak up though if you want,
Okay.
secondhand.
Thank you.
We're just street names, correct?
Two.
I'm gonna leave mine closed since yours is open because I'm afraid of it.
I have 2.
Are you guys done? You're just gonna, so to do, the next claim, the LB, you're gonna label that remaining, don't divide it. Take a loop full of string 1, and just kind of put it in the middle, sterilize, grab a loop full of string 2, and just mix it. You just want to encourage the 2 strings. So then you don't make the line. It's okay. But you want to label this as mating. So this is the plate that you're allowing the 2 strings to be in contact over the course of 40 hours. So what you do is make sure you label it and then grab a full string 1, put it in the middle, just like you just kind of do a little dab, sterilize your loop, grab a loop full of string 2, and then go over where you put string 1 and just mix it. You don't have to cover like a lawn play, you just kind of. Oh, so you heard. Yeah. That makes sense.
I don't even know if I did it this time. I don't know if I dipped it in or if I just took it out. I just did I cleaned it.
So the next thing is this. You're going to, what's your, you're going to label this as mating plate. You're going to grab a loopful, so sterilize your loop, grab a loopful of strain 1, put it in the middle, sterilize your loop, grab a loopful of strain 2, and just mix it into it. So you're allowing the 2 strains to come in contact with each other on the plate.
So one in the middle, sterilize, take 2 and just switch
Put it
it
on
around.
top?
Swish it around.
Okay, I'll
Yep. There's no, you don't have to cover the whole surface, you
Okay,
just want them
okay.
to come in contact. And then, Mimi, if, depending on how you guys divvy this up, someone also has to label the plates like initials and date.
Okay.
Once you guys, I think I talked to just about everyone, once you guys are done with all 5 plates, including the mating plate, make sure they have initials and date on them, and then put them in the incubator. Before you sit down, there's a handout on the middle bay too for the enterics.
Okay, so this one is, you're gonna label as mating.
Meeting?
Mm-hmm. You can label it anywhere. And you do not need this, so
Oh,
just disregard this.
okay.
No, it's okay. You're gonna sterilize your loop, grab a loop full of strain 1, put it in the middle, sterilize, grab a loop full of strain 2, and just kind of mix it throughout. So
Okay,
this is
so-
allowing the 2 strains to come in contact with each other.
Oh, okay, okay, okay.
There's no
Thank
perfect
you.
way to
Yep.
do this then.
I'm sorry?
There's no perfect way to do this then.
Correct.
Okay. What's today's date? 28.
My goggles are fogging up.
You put them in order.
I have them in order.
Well, you just go for it.
Well, this one for sure first.
LB, yeah. LB, yeah. They all go in the incubator?
Yep.
So we're done with the incubator?
Yeah, you can put it in the back.
First drink, so you just mix it.
For the one where you mix, you put the first one doing a circle, and then the other one just so they're in contact with each other. You don't have to like switch it like completely.
Oh, okay.
Just just. I like drag mine through a little bit and kind of just lit down.
Thank you. My goggles are like fogging. Whew.
All of them?
All those two, yes. I think everyone is at the same point, so I'll just explain how to Okay, so quick intro to enterics. If you've ever heard the term enteric, this is enteric-coated medicine. Enteric being relating to the intestines. So enterically coated medicine is medicine that survives your stomach until it gets to the intestines. That's where it's broken down and absorbed. Okay? But in this case, we're talking about enteric bacteria. So these are bacteria that exist in our intestines or intestines of vertebrates. Some are part of the resident microflora, some are pathogens. As a whole, they're gram-negative bacilli and facultative anaerobes. This is our second identification of an unknown. You guys should be wrapping up the one with the biochemical test or extracellular enzymes. This one is enteric unknown. We're going to carry out these 5 plates. TSA, which is just your general media, you can get the cultural characteristics from this. The other 4 are selective 4 gram-negative bacteria, which means gram-negative but not gram-positive should grow. And they are differential. Typically, this tells us whether or not they ferment lactose and produce hydrogen sulfide gas, but we'll get into that more on Thursday. There is some background information on that handout. Again, I'll get into that more on Thursday. At some point, I want you to prepare a street plate of each of these with your unknowns. So I handed out an unknown. Make sure you separate it from your E. coli strain 1 and 2. You should have a number that you're going to record. Okay? The other thing we need to do to identify your unknown is to prepare this analytical profile index 20E, also known as API 20E. And so let's start by doing that together. That is this test strip. This test strip is similar to performing all of those biochemical tests we did last week, just in a simple test strip. So think of each one of these as a biochemical test. We're going to add inoculum into these, what's called a cupule. We will look for a certain color change on Thursday. If it's one color change, that's a positive result due to enzyme production. If it's a different color change, that's a negative result, meaning no enzyme production. Okay? So first things first, we want to label everything. So you're going to label this. You can label right on the end here with your red wax crayon. And then you can also label on that plastic tray.
Down this. Yeah.
I put my initials and date, and if I can, I put my unknown number, just to make sure I don't lose track of it.
Oh, I
You
didn't
can't
see
write on the
this.
back of it?
You can try.
Yours is 51, mine's 30.
Okay. First step, it says is label, and then to fill the middle row of the tray with water. And so using your little squirt bottle,
Jesus.
with your distilled water, you're just going to fill this middle row. It doesn't have to be exact. That's just an approximation of how much water you should have. We do not want this to be filled with water so that when we add the test strip, it's floating in water.
You said
It's
just the
fill
middle?
that long strip.
You said the middle?
Our little bottles or this distilled water?
This one.
Oh, the whole middle row of water.
Yep, the whole middle row.
Oh, really? We don't have.
How did you go? You do have yours? Both of y'all have yours? It's like, I can't even take it off.
Yeah. Yep. Yep, you don't have the cover on it, right?
No.
Okay.
see the water, please?
Yeah.
You did it?
Okay? The next thing is it says using sterile technique, obtain a small amount of unknown from your, what will be your TSA slant, by just touching the loop to the growth and inoculate a tube of sterile saline and mix to dislodge the bacteria. So we are going to grab some inoculum from your known and inoculate this sterile saline. Now this is plastic, so we can't sterilize this. That reference to aseptic technique is simply referring to how you open this. So if you turn on your gas, just like always, you're going to sterilize your neck and then your tool. So sterilize your loop, sterilize the neck of the tube, and then when your tool is cool enough, grab some inoculum. And then just put it in this saline and kind of go back and forth to this one. So look, it doesn't
Maybe
really
turn this
matter.
on for me? No, I got it. So we grab number one. What are you guys doing? Grab this
Take,
one.
yeah, sterilize your tube, take, sterilize.
No.
Take some from it.
Make sure you're grabbing from your unknown and not from E. coli strain 1 or 2. When you're done, and I can demo this again for those of you that are still working, you're going to take a sterile transfer pipette and you're going to gently, like squeeze and release to make sure everything's well mixed. Once you feel like that inoculum is well distributed, you're then going to grab some and holding your test strip at a 45-degree angle, fill the cupules only to the bottom of the opening, not all the way, just the bottom of the opening.
So you don't wanna see it, basically.
You want to add it slowly to try to avoid bubbles as much as possible, but also try to avoid the
So when you
tip
do
of your
this, mix
transfer
it,
pipette coming in
squeeze it up
contact
in
with
and out.
anything in the cupful.
Make sure.
Because there's reagents in there and you don't want to
Can I
cause.
put it on the bottom one? Either bottom one?
No strip.
Just to the bottom of the cupful. Try to avoid cross-contamination,
No, that.
You're doing this for all of them?
all of them.
This one?
Nice and slow to avoid bubbles as best as you can.
And you're only filling it up into, not no spaces. Don't fill it up.
Can't see nothing.
Yep, looks good.
If we get a bubble, is that okay?
It's okay. Finish the whole thing and then see if you tap the strip on the desk, if that dislodges it.
So is it easiest? Is that too much or is that? Oh, I can't. That is
I thought,
too much.
okay, I thought you said silk.
No, just to the bottom. Keep going. Right there.
Oh, I see.
No, that's fine. And then when you're done, you can put it inside.
We'll just lay it.
Yeah.
And then I put the top on.
No, yeah, what did we start?
You said we lay it flat after?
See these two? Some of these, you wanna go to the bottom of it. This one's like perfect. Yes. I just don't put it right there. You know.
Why squeeze it back in?
Once you finish, you're gonna put the test strip in the tray on top of the water. Okay, and then it says, completely fill the cupules with inoculum, only in the test indicated with brackets. So you'll see that the test labeled CIT, BP, and GEL are the ones in brackets. And so with this test strip laying flat, I'm going to fill that cupule opening now. Remember, before, up to this point, you only come to the bottom of the opening. And so I'm going to add more inoculum so that it's completely full.
That are in brackets.
Yep.
Can we lay it flat on top of the water?
GEL.
I wanna make sure.
And then fill the opening to the bracket. So,
I got that.
you need help?
There's only 3 with a bracket.
Which one's the bracket?
Gel,
Citrus.
citrus, and BP. So those
Oh,
fill
okay. I
completely
see.
up.
Thank you.
The last step is, I think people have the mineral oil, the label up in, bumps off.
I know.
When it's in your squirt bottle. You're going to add that to the tests that are underlined. Fill it to the top. So there's a number of them, ADH, LDC, ODC, H2S, UR.
I think the ones that are underlined.
And what are we filling those with?
With mineral oil. That was, you have that clear squirt bottle to your left. That, yep. Fill the opening with that to the ones that are underlined. If you're
I'm
done
done
with the
with this
mineral oil.
These are the ones that are on.
Yes. Are you all done too?
Got me shaking.
There's none on the blue side.
Once you're done with that, you can start your street plates. So using your unknown from your TSA plate, prepare those 5 street plates. then you put the cover on. Yep. You might as well wait for your plate, so you can group them together.
And you're all set with this? Zach can go in bypass or anything? It's good. And this comes up here.
Why
Is
is the
this-
water underneath?
I think it needs some humidity to support its growth. Is this labeled in some way?
Yeah, the strip is.
Okay, you
Underneath.
might wanna try to put something here too, so that you don't have to look underneath where you could spill things. You know what I mean, when you get it on Thursday? This I'm gonna put in your bowl. You done with the oil too?
Now we just start. Wait. With the street plate, we're using what we got in front of us, the one before this?
The
Or
one
the
that has a
unknown.
number on it.
Okay.
Three to four downstairs.
No, I'm not on the back, so I always end up doing the wrong thing. I guess.
See, I would have charted without the numbers.
Yeah, I can't.
It
do
always
the
messes
numbers.
me
It
up.
messes me up too, I can't. I'll do the numbers last. Cause then when I turn it over. it's
Yeah,
all over
it gets
here.
like-
I don't know how to explain it.
Yeah, but then when you turn it over, you can still remember which one you did. That was number 4. The thing is,
We're doing this to all the street plates.
Yep.
Sterilize.
sterilize,
Then
inoculate.
turn on one maybe.
Step 4, you just don't, you don't.
This one.
she wrote them backwards like we do.
It's 1, 2, 4, 3. 1,
Yep.
2, 4, 1, 2, 3, 4.
Oh shit.
Yeah, it's definitely easier without the numbers. It's freaking hot. I usually bring all.
I'll charge my fucking fan too.
That would have been golden right now.
Once you're done, you can put your place in your test strip in the incubator, and then take off your PPE and I'll handle the dose. 5 plates and your test strip in the incubator. You can put the minimum oil back. Your unknown and your saline will come up here in the orange rack. The only thing left should be the E. coli strain 1 and 2. Just leave that on the blue rack.
You're done with your.
What do you put over there? These two?
Me too.
Forgot. No, I think it's up there. I'll probably take a picture after. And I'll send it to you.